Yeah, okay. Thank you for the introduction, and it's a great pleasure for me to stand in front of you and share some of the foundational knowledge that is important for everyone who wishes to plan and execute a base editor screen. So I will start off with the introduction slide just to put the base editors into the context. So there are a few well-known instruments that a screener can leverage in order to achieve the installation of the target mutations. And the first one is well-known and is called deep mutational scanning. Here, the open reading frames of the proteins are overexpressed, and the mutations can be installed at any amino acid position, and any desired mutations could be installed at a given position, which gives a full saturation potential to this technology. The second technology is a saturation genome editing. Here, users could define the area of interest that they would like to introduce the mutation towards with a guide RNA sequence, and after the nuclease introduces the cut to the gDNA, an exogenously delivered DNA repair template could be used to install a mutation. And the third technology is a base editing. So here we can also pinpoint the area of desired mutations based on the sequence of the guide RNA, but the mechanism of mutation installation is based on the nucleotide deamination. In a nutshell, base editing allows to turn A nucleotides into Gs and Cs into Ts. And last but not least is a prime editing. Prime editing is also Cas9 nuclease, Cas9-based method that allows to target regions of interest with the guide RNA sequence and install the mutations based on the reverse transcriptions of the extended sequence of the guide RNA. For the purpose of today's talk, I would like to specifically focus on the base editing technology. Brief historical overview: the first base editors were developed at around 2016-2017 here at the Broad, and since then multiple research groups have contributed to an enhancement and perfection of the technologies. Coupling the deaminase enzymes to the Cas9 nuclease with relaxed PAM preference allows to greatly expand the targeting scope in terms of number of the guide RNAs used for targeting. This slide just illustrates that since the arrival, base editors have been used to a great success in multiple research papers, which led to discoveries of new interesting answering many interesting biological questions. But regardless of the type of question that one might be interested in exploring, I would like to highlight two main types of screens, or type of signal screens, if you will. So there is an enrichment type of screen or positive selection screens where when the mutation arises, it gives a selective survival advantage to the cell in the population. So therefore, it leads to an exponential amplification of the mutation as the screen proceeds. And there is a second type of signal, which occurs in the depletion screens. So here the mutation leads to a cell death or stalling of the progress, and in this particular type of screens, the cells that harboring the mutations are progressively getting depleted from the population, which makes it particularly important to make sure that we have a good starting level of base editor signals in depletion screens. Another important dichotomy that I would like to introduce in the beginning of my talk is the difference between the genome-wide screens and the gene tiling screens. Genome-wide screens target multiple thousands of genes in the same screen, but they have a few guide RNAs which target each gene at a time. And these screens are particularly good for picking your initial hits or genes of interest that you would like to explore further. And in order to do so, the base editor tiling screens are proven to be a very useful instrument. Here we can target one or a few genes per library, but the density of tiling is very high, so we would have hundreds and thousands of guides targeting each particular gene of interest. The composition of the base editor library typically consists of two main groups. The first group is the gene of interest guides. So here the user must define which isoforms of a particular gene of interest one would like to target, whether they would like to cover 3' and 5' untranslated regions, whether they would like to include the splice sites of the gene, and how far they would like to crop into the intronic regions of a particular gene. And the second part of the guides in the library are the control genes. We recommend to allocate approximately 10% of the overall number of the guides in the library to controls, but up to 1,000 guide RNAs in total. The controls are also split in two subtypes. The positive controls, we recommend to have approximately 1% of the total controls, or up to 50 or 100 guide RNAs in total, which would target a splice site of the essential genes for a particular cell line. And the second group of controls are the negative controls. So here we would have approximately 9% of the total library size, or approximately up to 800 guide RNAs in total. So the negative controls could be also coming in two flavors. The first one is the guide RNAs which targeting the intergenic regions, and the second type of guide RNAs which would be targeting which would not have a predicted binding target within a given genome. So after you have decided which of the guide RNAs would be included in your final library composition, it is time to go to the lab and actually do the pipetting to prepare a library. We typically order our libraries as oligo chip synthesis from chip synthesis, and the composition of the oligo would contain the guide RNA sequence itself with the prepending gene nucleotide. The guide RNA sequence would be flanked by the restriction sites and also by the PCR primers for the PCR amplification. After the oligos have been PCR amplified, we perform the cloning of the oligos into the vector backbone, and afterwards the cloning product is electroporated to the electrocompetent E. coli cells and grown on the large ampicillin-resistant agar plates. It is important to take into consideration that a library representation should be maintained at this stage, and for this we find it particularly useful to perform the serial dilution of the library and a colony count to make sure that the representation is well maintained. And afterwards we advise to send the final plasmid library for the sequencing to ensure a balanced distribution of the guides in the library. After the plasmid library pool is ready, we produce the lentiviruses. For this, we use HEC293 cells, and we package our lentiviruses using a second generation of virus packaging system. A total duration of the virus production takes 3 days, and after the harvest, virus can be is filtered and can be stored at -80 degrees for extended periods of time before it is used. There are two main types of the library delivery approaches which we employ, and they are based on the type of the library backbone which is used for the library cloning. There is an approach that is called all-in-one vector. Here the base editor would be simultaneously co-delivered with the library of the guide RNAs. And the second approach is a split vector approach, where we first would establish the cell lines expressing the base editors, and afterwards these particular cell lines would be transduced with a library of the guide RNAs. So the positive things about the all-in-one vector approach is that it requires only one round of transduction and selection. However, the downside here is the large constructs tend to have a lower virus titer. And for the split vector approach, it would require 2 rounds of a transduction and selection. However, since the smaller constructs of the guide RNA library due to their smaller size, the titer of the virus would be higher. So it would be easier to achieve a good representation of the library at the transduction stage. There are few pre-screening optimizations that one would need to perform in order to achieve a successful base editor screen. And the first one optimization step that we recommend to perform is learning and establishing that the base editor has a good editing activity within your cells of interest. In order to check for the base editor activity, one may perform a base editor activity assay. Briefly, we would deliver the base editor into the cells of interest, perform a selection, and then we would deliver a second virus that is encoding the GFP protein sequence and expresses the guide RNA, which targets the GFP sequence. After the base editing occurs, it depends on the type of the editor. There might be missense or nonsense mutations introduced to the sequence of the GFP, which would then lead to a loss of the GFP expression. Then the assay may be read out using the flow cytometry or Sanger sequencing of the on-target editing itself. And the second important optimization step is that we would need to experimentally determine the optimal virus loading dose. So here, the main rationale for that is we aim to deliver only one guide RNA per each cell, which is very important for a successful execution of pooled high throughput screenings. And to do so, we transduce our cells at low multiplicity of infection, or MOI for short. MOI stands for the number of virus particles for each which end up in one cell. So in order to achieve good results in the screen, we would aim to have MOI of approximately 0.3. So how does one determine the MOI? This value can be extrapolated from the percentage of the infected cells in the population. However, as shown on this particular slide, the relationship between the MOI and a percentage of the infected cells is nonlinear, and one must always consult a special table which is presented to the far right of the slide here to extrapolate the MOI value from the transduction, from the percentage of the infected cells in the population. In order to determine the percentage of the transduced cells, of the infected cells in the population, we perform the inline titer assay. Briefly, we titrate the virus volumes, and afterwards we split the transduced cells into the two populations. The first one is the untreated one, and the other one is treated with a selection marker. In this case, is puromycin. After the puromycin selection is over, we calculate the ratio between the cells which survived the puromycin treatment versus the cells which were expanded in the untreated arm. And the ratio between the two gives us the percentage of the infected cells in the population which was achieved by a given virus volume. And from this, we can use this value to get the MOI. The following slide illustrates a typical timeline of the screen. So first, if we use the split vector approach, we would deliver the base editors and then perform a selection. And afterwards, we will transduce the cells with the guide RNA, or if it is the all-in-one approach, the base editor and the guide RNA library would go on the same day. And after 2 days, we start a puromycin selection, and it takes approximately 5 days to be fully established. And after that, we would collect our early time cell points, as well as if it is desired by the screener, the cells could be split into the drug and no drug arms. And we would follow up the cell pellet collections on day 14 and day 21. Here, it is also very much important to maintain a good representation of the library, and we tend to not let it drop below the 500x. After our final cell pellets are collected and the gDNA was extracted, we perform the PCR amplification to amplify the parts which contain the guide RNA sequencing. And for this, we use long primers which contain the on-target binding site represented in red here, as well as Illumina-compatible overhangs in green and blue. Our P7 primers contain the Illumina barcodes, which are then used for the demultiplexing of the samples. We perform our PCR reaction in 96-well plate format, and here it is also important to keep an eye on maintaining the representation of your guide RNA library, but it is also important not to overload the PCR reactions with a guide RNA with the genomic DNA material, since it might inhibit the reaction itself. On the same PCR plate as we load our gDNA samples, we also allocate a space for the plasmid DNA pool from the initial, which was used for the lentiviral, for preparing the lentiviruses. And here, this pDNA pool could be further used as one of the reference points for the screen analysis. This is not working. Okay. Okay. After reads are back from sequencing and demultiplexed, and we ensure that the sequencing data is good, it is time to do the analysis. So the first thing that we need to do is to normalize the row read counts for each guide RNA to values per million. This step is required since, due to various hands-on biases that might occur in the lab or technical differences in between the samples, the absolute read counts may vary from one condition to another, and bringing the conditions to reads per million helps to mitigate this this artificial, this bias. The second step that is required is log normalizing reads per million. This step is important since the next generation sequencing data tends to be right skewed. And what I mean by right skewed is the information is illustrated by the histogram in the middle. So some reads tend to be more represented than the others in the overall population. And in order to make our data amendable for the downstream statistical analysis, we would need to normalize the distribution, and this is why we resort to the log normalization procedure. Oh my God. Okay. Okay, after we got our log normalized value, it is time to calculate the log-fold changes. The log-fold changes are calculated by subtracting the log normalized values of the reference condition from our experimental condition. The resulting log-fold change value would tell us whether a particular guide RNA has depleted or enriched in the population. But in order to get the biological effect size for a particular guide RNA, we resort to calculating a z-score. So the z-score is calculated by subtraction of the mean of the log-fold changes of the negative control from the experimental condition, and then we divide this difference by the negative, by the standard deviation of the negative control group. Okay, so now you got your pretty Excel sheet with all the log-fold change values and z-scores in place, and it is time to dig in and understand how well your screen actually performed. The first metrics here to assess is whether your experiment is biologically reproducible, meaning how well your replicate A is correlated to the replicate B. And in order to do so, we need to understand which numerical values we should take for building such correlation plots. And here one important distinction that I would like to make is that if we use the log normalized reads from your Excel to perform this correlation, we might get a good correlation values even in the absence of a true biological signal in the screen. And to illustrate that, I would like to give an example. Here on the far left of the slide is the mock replicates which were constructed by introducing a degree of random noise to the reference condition, and then they were cross-correlated. And as we can see, they have a very good correlation coefficient. But if we bring the same data to the log-fold change values, meaning that we would compare them to the reference condition and then cross-correlate them, then the correlation would be much more we would see that there is no biological signal present in the screen. So this is illustrated by the correlation plot in the middle. Typically, when we are conducting the depletion screen and we have a good signal in the population, the correlation of the log-fold changes or z-scores would look something like is shown on the far right of the slide. Another important quality matrix for the screen is checking your controls. And by this, I mean that we would, if the screen was performed correctly and we have a good level of signal, one should see a depletion of the z-score in the positive controls, and the negative controls should not have a strong depletion or enrichment signal. So here on the far left of this slide, we see the depletion of the positive controls on the z-score axis, and if the screen did not work nicely, then we would see that the z-scores of the positive controls would cluster around the same as the negative controls. And if one would like to know how well a screen can distinguish between the positive controls in the library and the negative controls in the library, one could build a receiver operator curve where the true positives would be genes targeting the essential splice site, the splice sites of essential genes, and the false positive would be genes targeting intergenic regions. And as the better the screen can distinguish between the two, and afterwards we would need to calculate the area under the curve. So area under the curve of a good classifier, or the screen that performed well, would be close to 1 in its absolute value. And if the classification was close to random, then this value would be very close to 0.5. So the good screen is shown on the left-hand side, and the bad screen example is shown on the right-hand side. Finally, it is now time to zoom in into the performance of the guide RNAs that target your gene of interest. And here the important consideration relevant to the base editor themselves is that the base editor can edit nucleotides within a given editing window. So here is 4 to 8 nucleotide, and all the nucleotides that are amendable for a particular base editor would be edited. So on the amino acid level scale, that would mean that one guide can simultaneously introduce edits to multiple amino acids if they're adjacent to each other. So in the depiction on the right-hand side, we see that an example of the guide that introduces silence, missense, and nonsense mutation to the adjacent amino acids. And when we are at the stage when we would need to assign a z-score to the amino acid, it is important to understand that the z-score value of a particular guide is very likely to be determined by the most phenotypically severe predicted mutation. So in this case, it's going to be a nonsense mutation. Therefore, we can assign the z-score from the nonsense introducing guide to the amino acid 3 in this case, but we would not want to extrapolate this value to the amino acid 2 and 1, because this guide would, the z-score from this guide would be determined by the nonsense mutation. So we came up with a severity rank, which would be used in order to dissect this particular mutation categorization for the guides analyzed in our screen. And once your results are ready and you have z-scores assigned to a particular amino acid positions in your screen, the great visualization tool is also developed here in the Broad by Sumaya, and it's called G2P Portal, and it allows to transfer the z-score values from your skin of interest to the various features of your protein sequence. And this resource is great for visualization of the 3D structure of the proteins of interest. And last but not least, after every screen, it is important to perform a validation of the guides in arrayed manner and assess the exact editing which occurred on the genomic DNA level. This present slide illustrates the guide that was used for the targeting of the BCL2-like-1 gene, and it grants the mutation to a small molecule inhibitor treatments. So if we assess the gDNA level editing without a drug, we see an expected C to T editing within an editing window of the guide RNA. However, and a very small fraction of the unexpected CB editor editing patterns. And in the middle, once we would apply the small molecule treatment to a particular mutations introduced by this guide, we would see that due to the high selective pressure, the unexpected edits would progressively amplify in the screen and could also contribute to the resistance to a particular small molecule. And here, since we have a heterogeneous population, by scoring which of the amino acids were enriched and which not, we can dissect the causal mutations from so-called passenger mutations, and we could know which exact amino acid was responsible for the phenotype. And that concludes my talk. I would just like to shout out to a GPP platform here, the Broad. They're all contributing a great effort to performing screens at the Broad and with the collaborators at daily basis. And we're always happy to collaborate in designing and executing the screens of the high quality. Please do not hesitate to reach out, and yeah, I'm ready to take questions.
All right, well, thanks for a great overview of base editing screens on us. So maybe I'm gonna take chair's privilege and ask the first question. So one thing that we're nervous about, since we use lentis a lot to deliver libraries in other contexts, we've sort of developed maybe an unhealthy paranoia about lenti, like kind of residual plasmid DNA from the lenti prep creeping into the genomic DNA. And is that a problem that we should be nervous about? And do you see that? And how can we get around that?
So yes, it is definitely an important thing to consider. And in our group, we are currently working on a strategy which would allow to selectively digest the plasmid DNA from the lentiviral prep, the work led by Nathan. I know he was able to attend today, but it is definitely a consideration that one must take once planning and executing the base editor screen.
When you're doing your pooled oligosynthesis, is it better to have top and bottom strands synthesized and anneal them, or would you just do top strand and then do the second strand synthesis with a polymerase in-house?
So to my understanding, we are doing the single strand synthesis and then using the pairs of primers to selectively amplify from both sides.
Thank you. That was very informative. When you're preparing the plasmid libraries in bacteria, could you elaborate on why you grow them on large plates instead of just growing them in liquid culture?
Oh, absolutely. So once we grow our bacteria on the plates, the growth occurs in the localized colonies. And if there is a certain mutant or a certain type of a plasmid that, for example, was smaller or just have a selective growth advantage over the rest of the population, this particular advantageous mutant would be still restricted to a few colonies or like multiple colonies growing on the plate. And if we have a liquid culture media, this particular mutant have like unlimited space for the propagation, and it can very rapidly take over the population. So the agar plates are the way of restricting these particular outgrowing mutants to the local colonies.
Have you found that to be a problem in practice with liquid culture?
I have not practically executed the liquid colony versus the plate colony comparison myself, but this is like this is the predisposition that we use to actually opt for the agar plates.
Hi. Yeah, thanks for a really nice overview. Cheers. I was just wondering, because we often have issues of silencing of the base editors over time, so the editing starts high and then drops off. Do you have that issue? Do you have any ways you can get around it with the overexpression, and do you have to single cell clone every time? I was wondering what your success rates were, basically.
Yeah, so definitely a great question and a totally valid concern. So we have observed that if we keep our base editor off the selection marker pressure for the extended periods of time, the silencing of the base editor definitely occurs. And in order to mitigate that, we try for at least a split vector approach to time our transductions in the way that the library would go to the cells immediately after the end of the selection, and therefore we would hit our cells with the library and allow the editing to occur at the peak of the base editor expression. So this is a very good question.
Thank you.
I was wondering, what kind of tool would you recommend for the design of the guides for the base editing? And if I had, for example, 1,000 sites in different parts of the genome that I wanted to edit, is there a tool that could help me with designing the guides?
So we have an in-house script that allows for user to input the isoform of interest and define the features that one would wish to select as a base editor specification. So for example, if we want to use the NGG-PAM and GN-PAM, which editing window we would be presuming to use for our screens, and a few other features that would be important to consider. And this tool allows us to then receive the list of the guide RNAs that would be targeting a particular isoform of interest. And it is also designs the predicted mutation types. So for example, it would give you mutations for AB editors and CB editors. Please catch up with me after the talk. I can, in more detail, explain you how to access and use the tool.
Hi, great talk. I just wonder if you can compare base editing screen with prime editing screens, advantages and disadvantages.
Oh, definitely. So base editor screens have the advantage that they are fairly easy to execute and allow a dense tiling of the gene of interest. However, the downside of the base editor screens that we do not have a full control over the mutations. So we are restricted by the type of editor, AB or CB editor, to the types of mutations which would be occurring in our screen. And we are also restricted by the base editing window that we use in in our setting. For the prime editing, we can encode any type of demutations, and we're not restricted to the size, so we can also use the insertions using the prime editing tool. However, the downside of the prime editing screens is that it requires an extensive optimization to select and use the best PEG RNA or the guide RNA that is responsible for the editing in the prime editing setting. And if we are not careful in picking and optimizing this PEG RNA, we might hit the caveat that the guide RNA spacer is good, but the RTT PBS sites are not well optimized. So then the mutation types would be less. The mutation installation efficiency would be less productive. So there are pros and cons, and one must always balance the type of the editor that they choose for their screen based on the application. So in a nutshell, if you want a broad net to throw in the ocean and try to pull many things, the base editor is a great tool if you want to hone in and use a very optimized system and put time and effort into designing a well-performing PEG RNAs. Then the prime editing is also viable, is also a good approach.
Okay, well, great. Well, thank you so much for that great talk.
Thank you so much, guys.